Let's talk about some movement disorders. Let's talk about how the brain changes during learning. We now have multiple sources of knowledge about the role of the hippocampus in learning. On Canvas, there's a link that describes the London taxi driver study, which showed that taxi drivers in London who'd been driving the longest had larger hippocampal regions compared to other drivers. In this image, we see a PET scan of participants in a study in which they had to perform a memory task. The study showed that those who had greater activation in the hippocampus area actually performed better on the memory task. So, the more your hippocampus is working during a memory task, the better you will do. Research with animals has also shown that those species with good spatial memory have bigger hippocampi regions than species with poorer spatial memories. So these are species that either store food or do not store food. So birds and rodents that store food, they're essentially hiding the food, and they have to remember where they put it after the season is ended. So their memories about location are particularly good. When you have a species with good spatial memory, you find that that species on average have big hippocampal regions. Research with rats has shown that the hippocampus contains specialized cells that are involved in their spatial memory. Place cells are those that discharge when a rat is in a spatial location that is particularly relevant. Let's say there was food there last time and then the rat comes there again. Then the place cell can discharge, allowing them to make the connection between the location and the past event. There are also head direction cells in the hippocampus that discharge whenever a rat's head is in a particular direction. This comes in handy in making sure the rat knows not only where the particular location is in space, but also relevant to the position of the body. There are also grid cells in the hippocampus that discharge at many locations, forming a virtual grid that do not change depending on the rat's direction, movement, or speed. So they provide a layout of the environment. The place cells and head direction cells are located close together in the hippocampus. In contrast, the grid cells are found in the entorhinal cortex. It was the early work of Edward Tolman that came up with the idea that our brains create a cognitive map, a mental picture or image of a layout of our environment. His work was based on research with rats, in which he had rats solve mazes. And so when we see that even in humans, the hippocampus gets larger when people spend their lives driving taxis around busy cities, it brings to mind this research that started out with rats, that our brains are creating maps in our minds. In addition to the hippocampus, there are many areas of the brain that are involved in the storing of explicit memories. These are conscious memories that we can talk about. These areas include the amygdala, entorhinal cortex, parahippocampal cortex, pararhinal cortex, and the frontal cortex. These are shown in blue and purple in the image. Now let's take a look at brain changes in learning that occur at a low cellular level. It was Donald Hebb, the Canadian, who first postulated the Hebb rule, which described the fact that simultaneous activation of cells leads to pronounced increases in synaptic strength between those cells. So when learning occurs and cells are firing together during that activity, it actually strengthens their synaptic connections if it occurs again and again. So he came up with the phrase, cells that fire together, wire together, to describe this circumstance. Groups of neurons that become wired together because of their experiencing of the same phenomenon are called cell assemblies. Hebb proposed this hypothetical grouping of neurons that become functionally connected because of input. He believed that they played a role in perception, memory, and cognition. In the 1960s, the term long-term potentiation was proposed to explain the formation of long-term memories. It was first proposed by Terry Lomo, the Norwegian. Since then, it has been shown that glutamate, a neurotransmitter, is key to the process of storing long-term memories. When one blocks the glutamate receptors during a learning task, a researcher can prevent the animal from consolidating the memory or learning the task. That's all for now.